Hello guys, in this video I talk about Mendel's law of inheritance, which is an important topic in genetics. Right, so let's see. Let's talk about the Mendel. So Mendel was born in what is now a part of Czech Republic, and although his parents were simple farmers with little money, he was able to achieve sound education. A good education. Was admitted to Augustinian monastery in Brno in September 1843. After graduating from seminary, Mendel was ordained a priest. Of course, in a monastery, he was ordained as a priest and appointed as teaching position in a local school. So he became a teacher, and he was a good teacher. He excelled at teaching, and the both monastery recommended him for further study at the University of Vienna. So he attended the University of Vienna from 1851 to 1853. There, Mendel enrolled in the newly opened Physics Institute and took courses in mathematics, chemistry, entomology, paleontology, botany, plant physiology, all kind of things. And it was probably there that Mendel acquired knowledge of the scientific method, which he later applied so successfully in his genetic experiment. Now, it is important to note here that Mendel was the first. He was first to use something mathematical or statistical analysis into a biological experiment. Although nowadays it's very common to use mathematics or statistics in genetics, but in those days he was, I think, the first to do so. Now let's see Mendel's experiments. Mendel's first experiment explained the law of segregation and law of dominance. Right, so what he did was a monohybrid cross, in which he crossed two pea plants, Pisum sativum. One plant was homozygous tall and another plant was homozygous dwarf. So homozygous means both the alleles for the character were same. So if it's homozygous tall, then it will have TT, capital TT. So both the alleles, this T and this T, are same for the character of height. While homozygous dwarf plant, both alleles for short TT, it will have TT, a dwarf homozygous. Both the alleles will be same for dwarfness or for height. So here we can see what he really did in this cross. Of course, there is tall homozygous and there is dwarf homozygous, and they crossed it, and TT was the result, right? And this TT was tall. So it showed that the heterozygous plant was the F1 generation. This is the F1 generation, and all the plants in F1 generation were tall. But genetically, if we see, they were heterozygous tall, right? They were heterozygous tall because the two alleles, one is capital T, another is small t. So this is heterozygous, right? So it is a heterozygous. F1 generation was heterozygous tall. Now when selfing occurred, that is TT was crossed with TT, so we know how to form the Punnett square, right? So the phenotypic ratio for plants, tall plants to dwarf plants, was 3:1. 3:1 was the phenotypic ratio, that is 3 were tall and 1 were dwarf. While genotypic ratio was 1:2:1, that is capital TT, or the homozygous tall plant, was 1. Heterozygous tall plant were 2 and homozygous dwarf plant were 1. So the genotypic ratio was 1:2:1, while the phenotypic ratio was 3:1. Right, so by this experiment he gave two laws. The first was, of course, law of dominance. Mendel's law of dominance states that in a heterozygote, one trait will conceal the presence of another trait for the same characteristics, rather than both alleles contributing to a phenotype. The dominant allele will be expressed exclusively. So here we can see in the monohybrid cross that it did. This is a homozygous tall plant, this is a homozygous dwarf plant, and cross occurred, and all the F1 plants were this heterozygote. It was a heterozygote tall plant, and it was tall. So in phenotype, only capital T was seen, was expressed, right? The allele, that small t allele for the dwarfness of the plant, was not expressed at all. That means the capital T, which is allele for tallness of plant, conceals the presence of small t allele for dwarfness, and only capital T allele is being expressed in the phenotype. That means this capital T is dominant, right? While this T is recessive. So capital T for tallness is a dominant character. Now the second thing he observed was the law of segregation. So the law of segregation states that alleles segregate means distribute themselves. Alleles segregate randomly into gametes. When gametes are formed, each allele of one parent segregates randomly into the gametes such that half of the parent's gamete carry each allele. So in easy terms, what it means is that you can see in here, this is the parent and this is the parent, and when the gametes are formed, this T goes here, this T goes here, right? So they segregate, both these alleles are segregating. Similarly, this small t and this small t segregate or distribute themselves in one allele goes into randomly into one gamete, right? So that's all the law of segregation said, that the alleles segregate randomly into gametes, right? Also in the F2 generation in TT, right, in TT, this T segregate and goes into one gamete, this small t goes into another gamete. They segregate randomly and goes into a gamete. So that's all the law of segregation says. Mendel's dihybrid cross. So in the dihybrid cross, Mendel considered the inheritance of two characters together, right? So in monohybrid cross, Mendel had considered only one character inheritance. In dihybrid cross, he has now considered the inheritance of two characters together. Let's see, for example, he crossed two pea plants, one with homozygous round yellow seed and a homozygous wrinkled green seed. So here you can see capital Y, R. This is round and yellow seed, round for R and yellow for capital Y seed. And this is small y for green and small r for wrinkled seed. So these two types of seeded plants were crossed, and the gametes of course segregated separately. You can see capital Y and R and small y and R segregated, and in F1 generation all the seeds were yellow and round. That would mean that yellow and round characters are dominant characters, right? So they were the dominant characters, and these alleles also segregated and form all four types of gametes, capital Y, capital R. Here we can see more clearly. So you can see that the segregated capital Y, capital R, small y, small r, all segregated differently when selfing occurred in the F1 generation. Capital Y, capital R, capital Y, small r, small y, capital R, and small y, small r. So they it's actually independent assortment, and they segregated, and here we can see the ratio. Yellow and round were 9 yellow round seeds were found, 3 green round seeds were found, 3 yellow wrinkled seeds were found, and 1 green wrinkled seed were found. So the ratio, the phenotypic ratio become 9:3:3:1. That's right, this is the phenotypic ratio in the dihybrid cross. Now what it shows, Mendel used this experiment to explain the law of independent assortment. How? Let's see. The law of independent assortment. Mendel recognized in his dihybrid cross that the principle of independent assortment. This principle states that the alleles at different loci separate independently of one another. That means that the yellowness, the yellow character and the round character segregated which separated independently during the gamete formation, right? So of course that would mean that also the green and wrinkled characters separated independently of each other during the gamete formation. This is what is stated by the Mendel's principle of independent assortment, or Mendel's second law. So, but here we must remember that Mendel, when he proved the principle of independent assortment, he took two characters which are present at different loci and are physically apart. In fact, they are physically on two different chromosomes. The allele for yellow is on one chromosome, while allele for round character is on another chromosome. But if there was some physical linkage between these two genes, the linkage we would study in another video. What is linkage between two alleles? If they are closely packed on one chromosome, then they can be in fact inherited together. But here there was no linkage, so the pea plant showed independent assortment, that the two characters present at two different loci was segregated independently or separated independently. So in the next video we will discuss about numericals on these laws. Please like and subscribe the video. Thank you.